Now we are going to start about major functions of natural killer cells. First, starting about the things we have to know about their key roles. First, they kill human cells infected by viruses. And second, it produce interferon gamma to activate macrophages. Secondly, it is a member of innate immunity. It's member of innate immunity but its origin in lymphocyte. It is the only one cell of lymphocytic origin that belongs to innate immunity. Other all B cell, T cell that belong to adaptive immunity. It uses the perforin granzyme to induce the apoptosis of virally infected cells and tumor cells. You guys know in apoptosis there will be interferon stimulation by where there will be cytochrome C will be released and apoptosis will be done. And there will be activity enhanced by interleukin 2, 12, interferon alpha and beta, and produce interferon gamma that will activate the macrophages. And it induced to kill when exposed to non-specific activation signal. When it is going to stimulate, when it is getting any non-specific activation signal, but it is also having inhibitory signals that are MHC-I on target cell surface. Whenever the MHC class 1 is present on target cell surface, then it will be inhibited. Also killed via antibody-dependent cell-mediated cytotoxicity. What is antibody-dependent cell-mediated cytotoxicity? It means it needs an antibody to get activated. So the CD16 of natural killer cells go on to attach with immunoglobulin G, Fc region of immunoglobulin G, and it will be activated. Same like it is happening with eosinophils. Eosinophils also needs IgE to get activated. Okay, there are two cells that are going to activate antibody-dependent cell-mediated cytotoxicity, that are natural killer cells that need IgG that will attach with CD16, and second eosinophils that needed IgE. And the natural killer cells also having some explanations like how that markers are going and enhancing the thing. And thirdly, there is one more marker present in natural killer cells that are CD56. The CD56 also is called NCAM. The NCAM means neural cell adhesion molecule, and it expressed on the surface of natural killer cell. It is a useful marker. It is a useful marker for the natural killer cell. Secondly, it is these CD56 are going to found in brain and neuromuscular junctions. And secondly, is aids in binding to other cells. You already done with antibody-dependent cytotoxicity, in which there is activation of natural killer cells by IgG, and also there is activation of eosinophils by IgA. Secondly, MHC class 1 is the surface molecule of most of human cells, and it's present antigens to CD8. All you guys know CD8 T cells, and it activates the adaptive immunity against the intracellular pathogens. Some viruses are going, and you guys know the CD8 and MSC are going to attack against the viruses, and some viruses is going to block MSC class 1. When the natural killer cell find any cell having decreased the expression of MSC class 1, they go and kill that cell and destroy that cell. Why? The natural killer cell suspect that in that cell the virus is going to block MSC class 1 expressions. Therefore, they think that in that cell the viruses are going to grow. So they go and kill and destroy that. And simple, easy. At the natural killer cells, you guys know having the same lineage as a T cell and B cell, having which lymphocyte, and they don't join thymers, They having no memory and they don't require any antigen presentation by MSC. Now we are going to start about the major functions of B cells. The B cells are going to indicate by the humoral immunity. The humoral immunity is a type of adaptive immune response that involves the production of antibodies by B lymphocytes to neutralize and eliminate the extracellular pathogen and toxins. Understand? And they recognize and present the antigen and undergo the somatic hypermutation to optimize the antigen specificities. And second, they produce the antibody differentiate into plasma cells and to secrete the specific immunoglobulins. They maintain the immunological memory and their memory be persist and accelerate in the future response. And second, most important thing, they inactive their B cell. When they start to forming the antibody, they will become plasma cells. And plasma cells are going to produce different antibodies. Now we have to talk about the T cells. The T cells are part of adaptive immune system, that is a cell-mediated immunity. And secondly, there are millions of T cells in the human body, and each recognize a unique antigen via the T cell receptors. T cell receptors. And emerge from the thymus naive T cells. One day encounter antigen, it becomes the mature T cells. And key factor is there, the T cell only recognize peptide. They only recognize peptides. What is the antigen presentation to T cell? The T cell only recognize antigen when presented. For example, this is one microorganism and T cell is standing there. And it will not attack and nothing do with this microorganism because he's in his own attitude. He will say, first anyone come to me and give some presentation, so I will attack on that microorganism. So they the one said that antigen presenting cell will come, destroy it, make some pieces and make an antigen and go to T cell, present that antigen, and then it will become active and attack on microorganism. It's just like that. An antigen presentation produce peptide fragments on the surfaces and the major histocompatible complexes. The fragment placed on MHC molecule 1 or 2. If one, then it will attach with CD8, and if it will be 2, then it will go into attach with CD4. Remember the role of 8. And second, the fragment placed on MSC molecules and T cell react only to antigen when placed on self MSC, the MSC restrictions. The T cell receptor having two chains, alpha and the beta, and the alpha and beta chain that are surrounded by CD3 complex. And they have these CD3 complex are signaling complex that transmit the bound signal into the cell. And what are the T cell receptors? They are formed by similar process to antibody heavy chains, encoded by the gene that rearrange for the diversity. Already you guys know in the adaptive unity that VDJC, the variable, diversity, joining, and the constant. They are the hypervariable domains. They were different receptors for different microorganisms. They have two key subsets, the CD4 and CD8, and CD4 produce cytokines, activate other cells and direct immune response, and CD8 T cells are cytotoxic T cells. They kill the virus infected cells, also tumor cells. The CD4 T cells activated by antigen presenting cells or MSC class 2. And the antigen presenting cells can be dendritic cells. They can be macrophages, and they could be B cell. And MSC class 2 bind. The MSC class 2 bind with TCR and CD4 and expressed only on antigen presenting cell. Two protein chains are there, alpha and beta. They bind invariant chain in ER and prevent the binding intracellular proteins. They must merge with the acidified lysosomes. the MHC-2 are going to bind with TCR or CD4, and they are merging in acidified lysosomes. The peptide fragments in lysosome that invade chain release and antigen bind to MHC-2 and the surfaces. And how they will be co-active, we have to study next pages. First, we have to go just very repeat the CD4 T cell help P cells to make the antibodies and produce cytokines to recruit the phagocytes and activate other leukocytes. The CD8 plus T cell directly kill the virus infected and tumors via the perforin and granzyme, similar to natural killer cells. They having that type 4 hypersensitivity reaction, that delayed response, and also they have acute and chronic several organ rejection that can be given by T cells. Before moving to the next topic, we have to start a differentiation of T cell in a simple way. You guys know in the bone marrow, the T cell precursors will formed and they will go to the thymus. In thymus, there will be immature and mature T cells. The immature will be in cortex and mature in medulla. And immature in the cortex, there will be positive selection. The positive selection means that T cell having both mark as a CD4 positive also, CD8 positive also, plus they also having T cell receptors. The negative section is also there, in which they have to interact with their self-antigens. So, if the T cell is going to interact with its own antigen, then it will kill their own cells. That T cell will kill their own T cells. Then this T cell is not reliable and it will be destroyed or removed, or due to negative selection it will be eliminated from further process. So in negative selection, you only select that T cell that are not going to react with their own antigens. So further, what the thing happening, how the T cell converted into helper cells like that. It is so specific, the CD8 are going to convert in cytotoxic T cells. But the CD4 positive are going to convert in T helper cells. And you guys know there are two types, type 1 and type 2, and adult 2 also, T helper 17 and T regulatory. How did all we going to done? For example, this is T helper cell. If it have to make itself T helper cell 1, it have to stimulate by, it is going to stimulate by interleukin 12 and interferon gamma. If you want T helper cell 1 from T helper cells, so the T helper cell should be stimulated by interleukin 12 or interferon gamma. After that, they will be going to stimulate macrophages, and that macrophages are going to make granuloma formation and phagocytosis. Okay? And there is also, if this cell have to convert it into T helper cell 2, then it have to be stimulated by interleukin 4 or interleukin 10. If you need T helper cell 1, then you have to need interleukin 12 or interferon gamma. If you need T helper cell 2, then you have to need interleukin 4 and interleukin 10. Understand? Other types are T helper cells 70. But you guys not said what is the function of T helper cell 2. T helper 2 will go to B cell and then stimulate into plasma cells. And by that, there will be antibody production and hormonal immunity will be started. And by the T helper cell 17, you guys have to need tumor growth factor beta with interleukin 6. That will stimulate T helper cell to convert to T helper cell 17. And they have two neutrophil requirement. And also, when more T cell is there, that is called T regulatory cells. It inhibits the autoimmunity, autoimmune reactions. What it will have to do, it make a check and balance on all the T cells. If there anybody is not doing autoimmune reaction or not, and it inhibits all autoimmune reaction and protect our own body from our own immune system. And it is simple, the description of immune system and simple description how the differentiation T cell will happens. Now we will move to the diagram that is given by the book. Same like that in the bone marrow, there are the T cell precursors. And then we having the T cells in thymus and cortex, they are the immature. And the immature having both surface marker CD4 and CD8 both, and there will be a positive selection, then there will be negative selection in the medulla when they will become mature, and then they will go into lymph nodes. The CD8 become the cytotoxic and CD4 become helper cell. If the helper cell is going to convert into T helper cell 1, they need interferon gamma interleukin 10. And if it need healthy helper cell 1 to convert into 2, T helper cell 2, then it needs interleukin 2 and interleukin 4. And for the 17, it needs tumor growth factor beta, interleukin 1 and 16. And for regulatory, it needs tumor growth factor beta, interleukin 2. Let me make easy for you guys. For 17 and regulatory, remember tumor growth factor beta only. And for the T1 and T2, you remember these interferon gamma, interleukin 12 for 1, and interleukin 2 and 4 for Th2. But there is one thing more. When there is increase in interleukin 2 and 14, that will stimulate the formation of T helper cell 2, but inhibit the formation of T helper cell 1. And when there will be increase in concentration and interferon gamma and interleukin 12, then there will be formation of T helper cell 1. But it will going to inhibit T helper cell 2. It is so simple. And you guys already know the function of T helper cell 1 is active in macrophages. 2 will going to activate eosinophils, B cells, formed antibodies, and 17 are going to induce a neutrophilic infiltration, and regulatory will prevent autoimmune reaction and maintains the tolerance. The positive selection is going to happen in thymic cortex. It keeps the T cell that recognizes cell peptides to allow for cooperation in immune responses. The double positive thymus are expressing T cells that recognize cell peptides, MHC compressors receive a survival signal. So we're having both of them. And second, negative selection, it is happening in medulla, and it's removed the T cell that bind too strongly to cell peptide. If they are binding with their own peptides, then they will be removed. And the thymocyte is expressing TCR with a high affinity for self-antigen and apoptosis, or become regulatory T cells. The autoimmune regulations protein drive negative selection and deficiency lead to autoimmune poor endocrine syndrome, and that are already given. And they're having the mnemonic. Mnemonic is without air, your body will cheer. Without air, the body will cheer. Air means autoimmune regulators. And in this air means autoimmune regulators. And cheer means all the diseases that lead to autoimmune polyendocrine syndrome, like the chronic mucocutaneous candidiasis, hypoparathyroidism, adrenal insufficiency, and weaker candida infection. Sufficient lymphocytes are going to interact with each other. You guys already know that T helper cell is going to stimulate macrophages, and by that, interferon gamma. The interferon gamma is secreted by T helper cell when it enhances the ability of monocytes and the macrophages to kill the microbe by ingestion. This function is also enhanced by interaction of T cells, CD40 and CD40 on macrophages, and macrophages also activate lymphocyte via antigen presentations. Guys, these T helper cells are going to activate macrophages. T cells are going to stimulate with T cell. T helper cell 1, you're going to stimulate macrophages by secreting interferon gamma. And the macrophages, take this as one condition. Second, the macrophages also going to activate or interact with T cell by CD40 and CD40 ligands. And CD40 and CD40 ligand. 40 plus CD40 ligand. This is present T cell and that is present macrophages. And secondly, also the macrophages act as antigen presenting cell and go to active T cell. This is all the thing. It's a summary of that paragraph. How the T cell and macrophages are going to interact. The T cell is going to stimulate macrophages by interferon gamma, and macrophages and T cell interaction also done by CD40 and CD40 ligand, and macrophages also act as antigen presenting cell and going to active T cells. Now the cytotoxic T cells, they kill the viruses, neoplastic cells and donor graft cells by inducing the apoptosis by perforin and granzyme. Already you guys know. They release cytotoxic granule containing preformed proteins that perforin, granzyme B, and cytotoxic T cell from CD8, which bind to MHC class 1 and virus infected cells. Already you guys know that CD8 cells are going to form cytotoxic T cell that going to kill viruses, mainly synthetic neoplastic cells and donor graft cells also. Then what are regulatory T cells that maintain the autoimmune, stop the autoimmune reactions in the body. They help them maintain specific immune tolerance by suppressing it, CD4 plus and CD8 positive T cell effector function. They identify by the expression of CD3, 4, 25 and FOXP3. Activated regulatory T cells produce anti-inflammatory cytokines like interleukin 10 and tumor growth factor beta. This is a very important thing. What are anti-inflammatory? There are two types of cytokines, the pro-inflammatory and anti-inflammatory. Pro-inflammatory that induce the inflammation, and anti-inflammatory that stop the inflammation. So the regulatory T cells are going to produce anti-inflammatory cytokines to stop the inflammation and regulate and maintain the autoimmune reactions. That is interleukin 10 and tumor growth factor beta. The apex, the immune dysregulation and polyendocrinopathy, enteropathy, excellent. It is syndrome and that is genetic deficiency of FOXP3. You guys want to know the FOXP3? It's one expression that used for the identification of regulatory T cells, and that they will be high risk. The chance of autoimmune reaction will be increased, a high risk of autoimmunity. So they will be characterized by enteropathy, endocrinopathy, they will need dystrophy, dermatitis and other autoimmune dermatological condition, and is associated with diabetes in male infants. So it's a simple thing. The regulatory T cells are going to maintain immune system and stop the autoimmune reaction. They're going to identify by the expression of FOXP and other CD cell surface markers. The FOXP3 deficiency will lead to autoimmune reactions, and it's most commonly associated with diabetes in male infants. And they includes enteropathy, endocrinopathy, nail dystrophy, and other autoimmune dermatological conditions. We're going to talk about T cell and B cell activation. For activation of any of these cells, they need two signals to activate their self. Already they mentioned the antigen presenting cell, the B cell, dendritic cell, Langerhans cell, macrophages. Two signals are required for T cell activation and B cell activation and class switching. So first you have to learn about T cell activation. Which two signals are going to activate T cell? For example, in diagram they already explained first the antigen coming by the antigen presenting cell. This is going to MHC class 2 or 1, and this MHC2 class 1 is going to attach with their respective surface mark. For the 2 is going towards CD4 and 1 is going for CD8. This is our first signal. And also they attach with T cell receptor, TCR. And second signal is between the CD80 or 86, you already said B7 of antigen presenting cell with CD28, with CD28 of T cells. This is second signal. When this and these both signals are done in the T cell antigen presenting cell, then the T cell become active. Then the T cell become active. For activation of T cell, we need two signals. One is between MSC and CD markers. Second between CD80, 86 or B7 with CD28. The CD28 is present on T cell and CD4 and 8 also present on T cell. Understand? For B cell activation, we also need two signals. The first signal is just like that. MSC2 is going to attach. MSC2 is already on the B cell. It is going to attach with T cell receptors. As it is, as in T cell activation, the MSC is going to attach with CD of T cell. Okay, it may be 4 or 8. It is respective to MSC class 1 or 2. Okay, MSC on B cell. Then there is the second CD40. Their CD40 is going to attach with CD40 ligand present on T cell. The same mechanism you already ran in macrophages. Macrophages also having same surface marker as CD40. That is going to attach with CD40 ligand of C cells. Understand? For the cell activation, T cell are on the MSC2 for CD4, MSC class 1 for CD8. And they are going to attach. This is the first signal. The second signal is between CD80, 86 or B7 of antigen presenting cell that is going to attach with CD28 present on T cell. Same, here I am making B cell and here I am making T cell. In B cell there is already attached MSC class 2 that is going to interact with CD4 positive. And for second signal, the B cell having CD40 surface marker, they are going to attach with CD40 like by this the B cell will activate and there will be class switching. Class switching means the formation of immunoglobulin from one immunoglobulin changing of nature for this in the IgM is B cell is activated in producing IgM. Now after class switching it is going to produce IgE, IgG like that. That is class switching. Class switching also happening between the IgM and G and G to M also. So I think it is already understandable for you. And now our new topic, it is just activated by exposure to is antigen. The B and T cells become energetic when exposed to their antigen without co-stimulatory signals. Another example, peripheral tolerance mechanism. Sometimes the both signals are did not there, only one signal are there, like already you guys know, is only one signal. The first signal is just about the presenting of antigen, and second signal is about their own about CD8, 86P7 with CD28. If that signal is not there, then the T cell and B cell is not going to activate. And for B cell, there is CD40 ligand with CD40. And this is a energetic lymphocytes are unable to mount a normal immune system, such as proliferation, cytokine production, or cytotoxic activity upon encountering their specific antigen. And energy is a critical mechanism for maintaining self-tolerance. And often encountering self-antigens leading to downregulation of surface immunoglobulin receptors and desensitization of B cell receptors in signal pathway. And this is a simple mechanism of energy to prevent the self-tolerance and autoimmune reactions.